Hello guys, and welcome back to Excel Share Academy. So now we'll be starting with another type of the cytoskeleton protein, that is microtubules. Okay, so they are the second principal component of the cytoskeleton with a 25 nanometer in diameter. So who remembers the diameter of actin? Kitna tha? I told you it is important. The diameter of actin molecule was 7 nanometer. Okay, so now microtubule is 25 nanometer. Later, we'll talk about intermediate filaments, that is around 15 nanometer. And that's why it is known as intermediate filaments. Okay, वो बीच के filaments है. Okay, so like actin filaments, microtubules are dynamic structures which undergo continuous assembly and disassembly within the cell. So if you remember, in the case of the actin filaments, there was something called actin treadmilling. Right? So what was it? It just meant that here you have got the minus end and here the plus end. Correct. In the same way, microtubules also, they have got the plus end and the minus end. So here the new tubulin molecules will keep on adding and here they will get disassociated. Okay, so it was like a treadmill. So it keeps on, going on. Of course, we do have the capping proteins for microtubules also, so that it doesn't polymerize at that instant only. Otherwise, what will happen? Our chromosomes won't be separated properly, and how the organelles will move then, right? So we do have got the capping proteins to stabilize both the ends. Okay, now they function to determine the cell shape, participates in variety of cell movements, including some forms of the cell locomotion, the intracellular transport of the organelles, and the separation of the chromosome during mitosis. Okay, now you tell me from your understanding of like whatever you remember, during the mitosis, we know that at the metaphase, the chromosomes are aligned, correct? Here, let us say that two chromosomes are there, they are aligned in a cell. Now we know that here the centrosomes are there. Your microtubules goes like this, right? And each single sister chromatid are pulled in this direction. Am I correct? Right? Now tell me, this is your minus end, this is your plus end. So which part of microtubule is getting depolymerized? Keeping in mind that sister chromatids needs to be pulled towards the, pull apart so that there is a division happening. Is my question is clear? मुझे नाम लेके बोलना पड़ता है. Yes, Akshay, the question is clear?
Yes, ma'am.
Okay. So who would like try to give the answer? Shruti?
The addition happens on the plus end, right?
Yes. So this end, see, we know that microtubules will grow like this. Spindle fibers will come like this from the centrosomes, right? So if it is coming like this means what? The tubulin are added here like this.
Yeah.
The plus end. And you know that the plus end is the end which goes and binds to your chromosome. है ना? उसके बाद ये separate होंगे. So we'll come to this, like how the sister chromatids are, together. So there are certain proteins like securin separase. They help in the separation of the sister chromatids. So this will come here and this will go in that direction, so that the two daughter cells are going to have equal division of the chromosomes. Now the question is, ये तो plus end हो गया और minus end तो यहाँ पे है. तो कैसे क्रोमोसोम या सिस्टर क्रोमैटिड को आप इस डायरेक्शन में खींच रहे हो? So which end of the microtubule is getting depolymerized? See, खींचना मतलब क्या, यहाँ पे इसका लेंथ कम होना चाहिए ऑफ द माइक्रोट्यूबुल्स, राइट, and something to hold upon the cystochromatid so that this can come in this direction. It is not like a hand, like which can take a turn and towards the center. Yes, Prishita, you would like to try?
माइनस एंड,
बट अगर ये माइनस एंड से डीपोलिमराइज
यस
होगा, then
यस,
it will
डिटैच,
get, no, it will get shortened here. ऐसे छोटा हो जाएगा. The cystochromatids remains here only. ये यहाँ से डीपोलिमराइज होगा and it remains here only. There's nothing which is pulling them in this direction. तन्वी, अब तुम्हारी बारी है.
Ma'am, when it will depolymerize in the negative end, so it won't pull like in the direction towards negative.
कैसे
Like
कैसे
I just
करेगा? See, let us see like this. This is your microtubule. It is attached to your chromosome like this. ठीक है, ये यहाँ पे दूसरा वाला है. Now you guys are saying ये माइनस एंड हो गया, ये प्लस एंड हो गया. So this attachment is over here. ठीक है. Now you guys are saying that माइनस एंड से डीपोलिमराइज होगा. So डीपोलराइज होना मतलब क्या? ये धीरे धीरे, अरे इरेजर से तो पूरा ही चला गया. Anyways, हाँ. So what is happening slowly, slowly? This portion is getting removed, right? But it is not pulling the cystochromatid in this direction. Okay, I'll give you one hint. This is really very interesting phenomena. I'll give you a hint. There is a kid, okay? There is a small kid. वो बहुत जबरदस्ती मतलब like शैतानी करण whatever. Like what mother is gonna do? Going to pull his hand, okay, and drag him wherever their home is. Now imagine that scenario. Like there is a mom who is pulling the kid's hand and walking towards the home. Okay, let's say this is our home. Now what is happening is there is something called stochastic depolymerization which happens at the plus end. ठीक है, यानी कि usually at the plus end depolymerization doesn't happen. Okay, but what happens is that during microtubule, when the cystochromatids are supposed to be pulled apart, that time the microtubules just falls apart. Okay, so what is happening is that there are proteins, okay, there are proteins which is going to walk on the top of it. ठीक है, so these proteins they make hold of it. Let us say ऐसे उसने बना लिया है. ठीक है, now they start running towards the center, you know, 1 2 1 2 1 2 microtubules. So microtubules में जैसे भी हमने क्या देखा था, vesicle है, then they have got these proteins walking, right? Same with these proteins, instead of vesicles or any other organelle, they carry the chromosome and they start moving. Now what is happening is यहाँ से, like in Hindi we say ना पैरों तले जमीन खिसक गई, and the same thing is happening with this protein also. So the moment it starts running in this direction, this polymerized microtubules over here, it starts falling from here, and this protein will keep on running in this direction. तो जैसे जैसे आगे बढ़ रहे हैं उसके पीछे का ट्यूबुलिन मॉलिक्यूल्स डिसोसिएट हो रहे हैं. ओके, एंड एट मोस्ट टिल इट रीचेस इट्स होम. हुआ इमेजिनेशन लिटिल बिट. शुड आई एक्सप्लेन अगेन?
Yes, ma'am. One more time.
हाँ सॉरी, 3 लोगों ने एक साथ बोला सॉरी. शुड आई एक्सप्लेन अगेन, यस और नो?
Yeah.
ओके. So you have a chromosome here and here, okay? So your sister chromatids are actually wrapped around like this. And these proteins are removed with the help of the securin and separase, which we'll talk about it later, okay? Now, this is your microtubule, which is attached. Plus end ho gaya, ye minus end ho gaya. Okay. Now, there are motor proteins which walk on the microtubules. I'll make it like this. It's easy for me like this. So this motor protein is making hold of this sister chromatid. Same way, यहाँ पे भी दूसरा होगा. ठीक है? Now what is happening when this association is being broken of the cystochromatids? This cystochromatid will go in this direction. ये वाला इस direction में जाएगा, right? Now what is happening is that, as I said, stochastic depolymerization is happening at the plus end. So it's like, you know, falling of all the tubulin molecules. एकदम से वो नीचे गिरने लग जाएंगे, ठीक है? जैसे कार्टून में नहीं दिखाते, you have got the brick made street over here and the cartoon character is running and ये पूरा नीचे गिरता जा रहा है. Okay, so the same way here the protein, motor protein holding the sister chromatid, it's walking like this, you know, each step like this, one step. The moment it is keeping those steps, the tubulin molecules are falling off ऐसे, and there is this sister chromatid which is coming on the back. Okay, so just imagine this motor protein walking with this sister chromatid, and जैसे जैसे आपका ये motor protein आगे की तरफ बढ़ रहा है, वैसे वैसे पीछे से आपका tubulin molecules fall off हो रहे हैं. So at the end what will happen? No microtubules which is attached to your chromosome. Okay, I'll try to get a video. I'll try to get the video. Okay, I'll try to show. Okay, is it clear? Like thoda bahut to imagination mein aaya hoga, like how exactly this is happening? Little
Yes,
bit.
ma'am.
Yes, ma'am.
Okay. So before we proceed ahead, let's know like the microtubules are made up of what? Okay, so in the case of the actin filaments, it was only your G actin, globular actin, which polymerizes to become filamentous actin filaments, right? So usko hum F actin bolte the. In the case of the microtubules, there are two heterodimer proteins, okay? That is your alpha and beta. So alpha tubulin and beta tubulin come together. Now in this also, you can see that alpha tubulin is bound to your GTP. वहां पे क्या था G actin में? ATP. Okay. Now in the case of the tubulin, alpha GTP, beta tubulin, your GDP is there. Okay. Please do remember this is important. Now the inner diameter of your microtubule is 14 nanometer and the outer diameter is 25 nanometer. Okay. Now microtubules, with the help of this alpha and beta tubulin, can you see the arrangements over here? The arrangements are like the in the alpha helical regions. जैसे कि अगर आप सिर्फ blue को follow करो, right? It looks like as if they are arranged in the helical type, right? So all these together, they form a protofilaments, and together 13 protofilaments are there, which forms the microtubules. Okay, this is important. Please remember. I think I have another figure also. Yeah. Can you see it? So here we have got alpha beta tubulin and these are the arrays. So this particular line, if you see, this particular one line is known as one protofilament. Together like this, we have 13 and then your microtubule is formed. Okay. Yeah. So now coming to the structure and dynamic organization of your microtubules. So microtubules are composed of globular protein called tubulin. There are two types are there, alpha and beta. We also have got the gamma tubulin, which is concentrated in the centrosome. And we know that from the centrosome, the microtubule starts, right? So tubulin dimers polymerize to form microtubules, which generally consist of 13 linear protofilaments assembled around a hollow core. and that's why it has got the inner diameter and the outer diameter. ठीक है? Okay, so the protofilaments, which are composed of head to tail arrays of tubulin dimers, are arranged in parallel. So microtubules, like actin filaments, they are polar structures, and that's why they have got the two distinct ends, plus and minus end. The polarity determines the direction of the molecular movement of the motor proteins, just as in the case of the actin filaments, जो myosin के लिए था. ठीक है? The same way here also we have got for the microtubules motor proteins. So again the same thing. So we have got the plus end, sorry, and the minus end. Okay. Now this is how the protofilaments, okay, of the microtubule starts depolymerizing. All the 13 protofilaments will start depolymerizing. It will look as if, you know, the flowers is there and it is shedding off. Now, microtubules are highly dynamic structures that can undergo rapid cycles of assembly and disassembly. Both alpha and beta tubulin, they bind to the GTP, which functions analogously to the ATP bound to the actin. So every time it is like, you know, compared with the actin. Now the GTP bound to beta tubulin is hydrolyzed to GDP shortly after polymerization. GTP hydrolysis weakens the binding affinity of tubulin dimers for each other, which can lead to rapid dissociation of GDP bound tubulin from each ends of the microtubule. Same thing. जैसे एक्टिन में आपका ATP था, वहां पे ATP हाइड्रोलिसिस के बाद ADP बनता है. That's how the depolymerization starts. Here also it's the same thing. Okay, yeah. So look at over here. So the role of GTP in microtubule polymerization. So when we have got at the plus end, so GTP bound tubulin is being taken and they keep on adding, right? And the moment the hydrolysis is happening, what is happening? The depolymerization will start. Okay, clear? So please remember here, the GTP which is bound to the beta tubulin is getting hydrolyzed. Okay, this is another one. So we have got here the arrangement. As you can see, there is a capping protein. Now why this capping protein is required? Onto which your microtubules will form. So we need certain base. Like for example, in the case of the actin filaments, we needed which protein? The base protein. Anyone who remembers? Formin, right? Onto the formin dimer, your first 3 G-actin comes and forms the nucleation, right? In the same way here, the capping proteins are there. First, your gamma tubulin will come. On to top of that, your alpha and beta tubulin will bind. Okay? Now, when these capping proteins are not present, this particular thing is not present, then only the depolymerization starts from this end. Okay? Now, as we see, so we have got a growing microtubule. So GTP bound, those are coming and getting bind and it is getting polymerized, right? Now it is in this stage. Now, if you have a microtubule which at the plus end itself is depolymerizing, can you see it? The same thing, okay? It is also known as the shrinking microtubule. That is catastrophe. That is what I was talking about, like, you know, all of a sudden catastrophic microtubule depolymerization is happening. So this kind of depolymerization is happening during the chromosome separation. Okay, did you get it? Please remember this figure. Okay, yeah. So, because of the central role of microtubules in mitosis, drugs which affect microtubule assembly are useful not only as experimental tools but also for the treatment of cancer. So that time, they, you know, many people who were working on this, they found that colchicine and colchicine, they were very good drugs which inhibits the microtubule polymerization. जिसकी वजह से माइटोसिस ही नहीं होता था. तो इनिशियली दे स्टार्टेड यूजिंग colchicine and colchicine as the anti-cancer drugs. Okay, then another ones are also there. So colchicine we use in the lab just to see how the metaphase anaphase is looking, right? In the lab you must have used. Now the other two drugs are there, vincristine and vinblastine. So this they have extracted from the flower. And what is the flower? Periwinkle flower, yeah. In Hindi we say that's सदाबहार flower, जो लोग ऐसे चाय वाय में डाल के भी पीते हैं in India. Okay, so they came to know that these molecules are able to inhibit the rapidly dividing cells by blocking the mitosis. Okay, the other drugs are there, Taxol, again which stabilizes the microtubules. यानी कि stabilizing the microtubules means it is not allowing the depolymerization. अब depolymerization नहीं होगा तो क्या होगा? Chromosome separate नहीं होंगे. Again, the mitosis is being blocked. Okay, so all this stabilization will block the cell division, and Taxol was used as an anti-cancer agent. Yeah. So this is the table which you guys have to remember, because in day-to-day lab, when whoever is working in the cancer region, okay, related to cancer, they should know how colchicine, paclitaxel, vinblast, and vincristine, all these drugs are actually acting. Okay, so here we can see colchicine, it inhibits polymerization via microtubule depolymerization. Then demicolchicine, again polymerization. Nocodazole, polymerization. Paclitaxel, it is stimulating your polymerization. Then vinblastine and vincristine inhibits polymerization. Vinorelbine inhibits polymerization. Okay, now when you say via tubulin crystallization means what? It is blocking the addition of the tubulins. Okay. Yeah. Now, let's come and let's talk about the microtubule associated proteins. Okay. Now, they are responsible for regulating the dynamics of the microtubules, like kahan pe polymerize hona chahiye, kahan pe depolymerize hona chahiye. So these all are decided by the microtubule associated proteins. It is very important to remember about the microtubule associated proteins because they are also involved in your cell cycle. ठीक है, यही same protein दोबारा हम cell cycle में भी पढ़ेंगे. So the minus ends of microtubules are stabilized by proteins which will prevent the depolymerization. Now, the growth or the shrinkage of the plus end of microtubule is regulated by MAPS, either polymerization or depolymerization of the tubulin dimers. So depending on, like अभी जैसे कि for example मुझे chromosome को separate करना है, we need to take these chromosomes into the different poles, right? Then I need that depolymerization should be happening at the plus end of the tubulin, correct? So that will be again regulated by the MAPS. Okay, like see, you have got the growth where normally the GTP bound tubulins are coming and adding. Okay, so the polymerase. Now, why we are saying polymerase? Because MAPS are your polymerase, which will help in binding to the plus ends of the microtubules, helping in the growth of the microtubules. Okay, so MAPS are actually the polymerase. So here, if you see the polymerase, it keeps on adding your newly introduce GTP bound tubulins. So this is in the normal case how microtubules are growing. Now suppose depolymerization has to start. Then the depolymerase will come. It will initiate the GTP hydrolysis, and what will happen then? Then the dissociation will start. तो इसने क्या किया depolymerase ने यहाँ आते ही उसने signal दे दिया, okay, I need depolymerization. So all the GTP bound tubulins molecules, they will get hydrolyzed. ये पूरा का पूरा आपका भी GDP बाउंड हो जाएगा and it will result in the complete depolymerization. ठीक है. Now, the another class of microtubule associated proteins are known as the CLAPS proteins. They act to suppress microtubule catastrophe and promote rescue, apparently by stopping the disassembly of microtubules and restarting their assembly. इसका मतलब क्या? For some reasons or maybe some because of the addition of certain drugs, what happens? The microtubules will start dissociating, and I need to stop that unwanted or unregulated dissociation, and that will be done by the CLAPS proteins, which will act as the rescue. Okay, and that will be prevented and restarting the assembly. Okay, for example, here all of a sudden this GDP bound catastrophic depolymerization is happening. So this class protein will come, it will hold onto the depolymerized state of the tubulin and will help in the growth of the further by bringing GTP bound tubulin molecules. Did you get it? So these are the three ways by which microtubules can grow and shrink. तो पहला हो गया जहाँ पे maps are coming acting as a polymerase where they are binding the GTP bound tubulin molecules. Second में क्या हो रहा है? आपका depolymerization हो रहा है, ठीक है? Now just imagine this shrinkage or the catastrophic depolymerization is happening. Then these collapsed proteins will come and help in the rescue. तो वो क्या करेगा? इसको dissociate नहीं होने देगा. Since it is GDP bound, it should be dissociated. But with the help of the collapsed protein, these will not get dissociated. It is prevented. And onto this, using as a base, more new GTP bound tubulins will be recruited for the polymerization. Yes. Any doubt? Is it clear? Microtubules. So today we talked about microtubules, like what are the tubulin, alpha, beta tubulin, gamma tubulin we talked about. We know that now the GTP is involved. We have seen that it is also having the dynamic stability or instability like actin filaments where both plus and minus ends are there for the depolymerization and polymerization, right? Okay.